Okay, good evening everyone. We'll be starting the session just now. Mhm. Okay, so welcome everyone to the week 4 of the PMRF-NPTEL session on Introduction to Developmental Biology. These are PMRF-NPTEL problem solving sessions in which we go through the quizzes of the last year's run of this course. So we better understand the course material as well as how to answer the questions that are asked to us in the exam, as well as the weekly quizzes of this year's course. So I will briefly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow and PhD scholar at the Department of Biological Sciences and Bioengineering, IIT Kanpur, and I will be taking this course. So how we do this course is that we go through various questions of the previous year's run of the course, and I ask you the question and give you some time to try and figure out what the answer is, and then we discuss the concepts behind the question, and then we answer the question and see if we were right or what exactly did we need to do to solve the question. So this week we have learned a lot about how genetic uh, methods work and how exactly we can go from identifying a particular phenotype all the way to the gene, as well as learned via examples how they were done historically in case of C. elegans. So this is what we are going to deal with today, and let us just get on with it. So the first question is, which of the following was discovered while studying cell lineage in C. elegans? So this image here is a hint. This is John Sultans, who actually did all the lineage tracing in C. elegans. And the options that you have are that different germ layers give rise to different organs. Genetic regulation in cells gives rise to different cell types. Certain cells are destined to die during embryogenesis, that is apoptosis, and environment determines the developmental potential of the cells. So these are the four options, and you can choose whichever you think is correct. We'll give you a moment for it. Yes, okay. So let us look at it. So what exactly is lineage tracing first? Lineage tracing is the experimental process of labeling a cell in a developing organism and tracking the descendants of the cell through subsequent divisions and differentiation to determine what tissue or cell types eventually become or like come from these particular cells. So as you would know that all life starts with a zygote, which is just one cell which divides and then forms the entire adult body. So how this works is that during these cell divisions, the cells go rounds of differentiation or maturation, and once they keep on dividing, they lose this ability to produce more and more cells and become much more specialized. So in the developing embryo, you will find multiple precursor cells or primordial cell layers which actually give rise to the final structure, and most of the descendants of these precursor cells actually form part of organs, and if this particular cell was not there, then maybe this structure does not form. For example, there have been multiple studies in the retina in which they have shown that the retina is composed of 7 different layers, and each primordial cell that actually divides gives rise to all the different layers in a sequential manner. So one cell will give rise to all the cells sequentially in that layer as it undergoes more and more cell divisions. So this is something that happens during development, and a very interesting way to find out what cell produces what or tissue, or what part of a tissue is known as a lineage tracing. There have been multiple methods historically where people have done it, but nowadays what we use is the loxP system. I will just give you a brief description of how the loxP system works. So Cre is a particular recombinase protein which can do specific changes to the DNA. So it is a recombinase, so it can cut, paste, do stuff to the DNA, and it recognizes loxP sites. So together they are known as the loxP system. How it works is that, say you want to trace a particular lineage of cells which you know are marked or express a particular protein, say PRDX1. This the entire mesenchymal lineage of the limbs will always be expressing PRDX1. So what do? You make sure that the organism has Cre under the PRDX1 enhancer. So whenever PRDX1 is expressed, Cre will also be expressed because it also has the same enhancer. So whenever the cells decide that this is the time I want to express PRDX1, or whichever cells are expressing PRDX1, they will also start producing the Cre protein. Now this is known as the driver. So the Cre as well as the tissue specific enhancer is known as the driver, and this is normally present in a one of your mice. So most of these experiments are done in mice. So you have a driver line with the enhancer, with the Cre present in its genome, expressing throughout the like all over the organism. But the Cre will only be expressed in the cells which start expressing PRDX1, which is the mesenchymal lineage. Next, you have a slave line or a driven line, in which what you do, you produce particular changes in the genomic DNA. So your gene of interest, whenever PRDX1 is expressed, you have a particular reporter such as GFP under the control of a very strong enhancer such as ROSA26. So in the driven line, you have ROSA26 followed by GFP. So when it will always start expressing GFP, but that is not what you want. So what you do is that you place a stop cassette before the GFP. So essentially ROSA26 wants to start expressing the cells, expressing the GFP, but since there is a loxP around, there is a stop cassette before the GFP, it never gets expressed. That is the driven line, never expresses GFP. But when you make these driver, the one that expresses tissue specific Cre, and the driven line which expresses GFP but is not able to because of the stop cassette, when they come together, what happens is that Cre is expressed only tissue specifically, say in the example PRDX1, only in the mesenchymal lineage cells. So Cre is expressed, and what Cre does is that it makes a cut at the loxP set as shown right here. So in green right here is our loxP, and this red thing here is our stop cassette. So in the cell specific expression, whenever Cre is being expressed in the cells where PRDX1 is being expressed, so the mesenchymal cells that you were tracing, Cre is being expressed, it goes in and cuts up the DNA in between the two loxP sites. So this makes a DNA loop and then goes out. Once the stop cassette is removed, now the ROSA26 enhancer can start expressing our reporter protein, which is GFP. So all the cells which expressed PRDX1 will start to have GFP in them, and since it is present in the DNA sequence, they will always express GFP, and this, using this, you can start tracing where exactly these PRDX cells go, and if they further differentiate down the line into other cells, say cartilage or bone or anything else, all of these will have GFP in them. So with this particular technique, you can figure out this is my cell and want to know what it makes. So you just have to find a particular gene that only this particular cell expresses, and you then trace it all the way to maybe birth or maybe adult stage even by using this technique. So this is an example where particular cells were being lineage traced, these are probably the somatic cells, I believe, and they and you can trace them all the way to the adult organism and see which tissues arise from these particular cells. So this is known as lineage tracing. So in complex organisms, this is one of the methods you can do it. But this person here, known as John Sulston, he did it all the way for C. elegans. So C. elegans, as you would know, is a very small nematode which has an exact number of cells always. So 959 cells is what it has, and it has invariant cell division during development. So always all the cells divide the same way in all organisms. So it is not like ours where some cells, if they are not there, or like some cells, like the number of cells is not absolutely certain throughout development. But in C. elegans, it is always known that what stage what number of cells will be there. So this is one of the lineage traced maps that he had created, in which he is tracing all the way from the P0 zygote to different cells as they are dividing. So, and during this, what John Sulston discovered is that some cells of the 959 cells that are there during development, or they always form, but then they die off. So as development proceeds, these cells will be formed, and these particular cells, if you trace them from their birth to adulthood, you will see that these particular cells are always known to apoptosize or die, as if they are being programmed that, yeah, your work is done, you do not need to be there anymore. So programmed cell death during development was what John Sulston figured out. And if you are thinking that this is something very specific to something like a nematode, that is not true. In the previous examples that we have seen in previous weeks, is that our hand, as it develops during like early embryonic development, it is actually something like a bud. So it is like a spherical structure which finally forms our fingers. But all the spherical structure has cells in between the fingers, which is known as the interdigital mesenchyme. All these cells, in case of us, actually apoptosize and make sure that our fingers are free, and that is why we have a separate hands. But in case of other organisms such as bats, there the interdigital mesenchyme does not die off and instead forms the wing of the bat. So apoptosis is something that was discovered in nematodes and occurs throughout development in many organisms, and it is not specific to just nematodes. So this is what John Sulston figured out during his study of cell lineages in C. elegans. So the correct option is certain cells are destined to die during embryogenesis. Okay. So if that is okay, we move on to the next question. The next question is, ethyl methane sulfonate is commonly used as a dash in genetic screens. The options are a DNase inhibitor, a proliferation inhibitor, a mutagen, or an oncogene. So take your time, try and figure out what the answer is. Okay. So what is ethyl methyl sulfonate, or EMS? EMS is a particular mutagen which, when introduced to cells, causes different mutations in the cells. But people during mutagenesis screens have figured out a very smart way to use it. When they put in a low concentration of EMS for a low amount of time, they can make sure that there is only one mutation in a particular locus throughout the genome. So they can make a single mutant, or a single where a particular gene is mutated by the use of EMS. But why do we need that? These are used in case of mutagenesis screens. So early on, when the field people were still figuring out the phenotype to genotype relationship. They didn't know which genes functioned, like what the function of different genes were, and they wanted to figure out how exactly these genes make the entire organism. So the idea was very similar to, say, if you were going into a fully lit hallway. So if there is a fully lit hallway and what you want to know is that, and you have a switchboard in front of you, you want to know which switch controls which light. So how do you do it? You do it, like, the most obvious answer is that you start switching off the lights and see which particular light goes off when you turn off a switch. So that switch must be powering that particular light. The idea is very similar here. Whenever there is a EMS induced mutation in the genome, where if the mutation lies on a gene, then probably the gene is not going to function anymore. So you are going to have a mutant loss of function allele. And when you let that embryo develop, it will probably show some kind of phenotype which is different from the wild type. So you will be able to figure out, yes, then this particular uh, mutant actually shows a phenotype. So whatever gene I mutated first with EMS actually has a role towards development. And then they did a lot of things to figure out what exactly that gene was, and this is how most genes and their functions were discovered. The most famous example is Christian Nuslin Volhard's screen on Drosophila, where she figured out like a ton of genes and their functions in development. So we are probably going to come across it in further weeks. So this is an example of how exactly you do a mutagenesis screen. So you have a normal C. elegans hermaphrodite, and what you do is that you put in EMS. So you have you have a worm, you put it in EMS, then you self cross it. Whenever you self cross it, then it will have, if there is a mutation, then the genotype should be M over positive, but since it has a wild type copy, the phenotype might not be obvious, because the wild type copy is going to be, if the situation is such that the wild type copy recovers the mutation because it already has a wild type allele, then you will not see a phenotype here. You self it again, and this is what you should see in the F2 generation. You should have a wild type over wild type worm, you should have a mutant over wild type, and you should have a worm which has both the mutation in both the alleles. This is where you will be looking for your phenotype. So in a plate of worms, you will look for a phenotype, and this is the worm that will show you the phenotype. And then you go after it with all your genetic tools and figure out what exactly was the mutation and what was the function. We have followed an example like this in the course, where we have seen vulva development in C. elegans and figured out how exactly it works using just these mutagenesis screens. And one more thing to keep in mind is that, say you went after this worm, you figured out everything you needed to know by making a live set out of it, doing your northern blots, western blots, everything else, but this particular worm is now done for. Like, you have utilized everything it had to do your experiments. So how will you do further experiments? Like, how will you figure out genetic crosses from this? So the idea here is that you use you keep the F1 progeny alive, and you can always make sure do these self crosses again to again form another series of double mutant worms that you can then do experiments on. So this is your stock of worms that you keep to make sure that you always have these worms in hand, and these are the ones you do your experiments on. So ethyl methane sulfonate is commonly used as a mutagen in genetic screens. Okay, then if this is fine, then we move on to the next question. The next question is, what is the key difference between 2-factor and 3-factor analysis of genes? The options are: 2-factor analysis uses 2 reference alleles, while 3-factor analysis uses 3 alleles. Two-factor analysis can be used to determine the chromosome on which the gene of interest is located on, while two-factor analysis provides information on the nature of the mutation. Two-factor analysis can only identify gain of function mutants, while three-factor analysis can distinguish between gain and loss of mutants. Yeah. Two-factor analysis can be used to determine the chromosome interval or the chromosome on which gene of interest is located on, while three-factor analysis identifies the genetic interval on a specific chromosome in which the gene of interest is located. Two-factor analysis can be performed at F2 generation, while three-factor analysis is performed at F3 generation. So I will give you a moment to try and figure this out. Okay, so let us look at it. So what is two-factor and three-factor analysis? So two-factor and three-factor analysis are used in genetic mapping, and the concept it is based on is how tightly two genes are linked to each other. So if you remember the last week's course material, whenever there is meiosis occurring, there is a chance for recombination to occur. But if two genes are present very close to each other, they are known as tightly linked to each other. So there is a possibility that when they are in one of the genes is inherited, then both of them will be both of those alleles will be inherited at the same time, because they are really close to each other, and there is a very low possibility of being a recombination between those two genes. So these are known as linked genes. So two-factor analysis, in two-factor analysis, you figure out the recombination frequency between the new mutant and known marker mutation, which tells you, yes, my new mutant is around this particular marker mutation, because both of them are linked and inherited together. But if you have created a mutation and you want to figure out, so if you have done a mutagenesis screen and want to figure out what exactly the gene is, then you need to figure out where exactly the gene is. So to do that, you do something known as three-factor analysis. So what you do, you figure out the recombination frequency of the new mutant allele along with two different markers. So in this example, say our mutation was in CN, and black and vestigial, these two are our known markers. So what you do is that you figure out the recombination frequency between black and CN, which comes down to 9%, CN and vestigial, which comes down to 9.5%, and you know between black and vestigial it is 17%, because they are known genes and known markers. So using this map, you can figure out that our mutation lies somewhere between the black and the vestigial locus, and thus you are able to figure out where in the entire genome your particular mutation is present. This is a very powerful tool to actually shorten the interval of the genomic sequence to figure out where exactly your gene is located. So what you will do is that you will start off with markers which are present on separate chromosomes, so 1, 2, 3, 4, 5, and then you figure out, yes, my gene is being independently inherited compared to markers on chromosome 1 but not with 2, and then independently inherited with 3, 4 and 5. So if they are independently inherited, meaning they are too far away from our marker, so maybe they are present on the second chromosome. Then you take two known markers of second chromosome and then do the recombination frequency analysis again, and you will figure out that yes, they are if you get something like this where CN is somewhat like the distance between the two, the CN mutation lies between black and vestigial or any other markers that you are using, then you know that this is the stretch where I have to look for my mutation. It could also be that CN was present all over here. In that case, you will see that vestigial and CN, the recombination frequency is say 5%, black and CN, the recombination frequency is 15%, and you know that black and vestigial has a recombination frequency of 17%. Since that just shows that it is close to vestigial but away from black and not present in between the two of them. So you know that it is somewhere towards the right of vestigial, so you figure out another marker which is towards the right of vestigial, say somewhere over here, and do the recombination frequency analysis again till you are able to figure out two markers in between which the mutation lies, and that is where you should look for the mutation. So this is what you do with three-factor analysis. With two-factor analysis, you can figure out where the gene is. With three-factor analysis, you can narrow it down to a particular locus or particular segment of the genome which you can actually probe with molecular biology or other genetic techniques. Okay, what is the key difference between two-factor and three-factor analysis? The answer is two-factor analysis can be used to determine the chromosome on which the gene of interest is located on, while three-factor analysis identifies the specific genetic interval on a specific chromosome in which the gene of interest is located. Okay, if that is all for this, then we move on to the next question. On a chromosome, we situate three mutant alleles of three different genes A, B and C to study the recombination frequency among them. We observe that the recombination frequency between A and B is 26%, while the recombination frequency between B and C is 10%. Additionally, the recombination frequency between A and C is 18%. So which of the following inferences is the correct one? So all the options are like, where is C as compared to A and B? So C is between A and B and closer to A is between C and B, closer to C, B is between A and C, closer to C, and everything else. So I would like you to take your time, go through the option, because this is exactly what the three-factor analysis we just discussed is. So try and figure out what you think is the correct option. Okay, so let us look at it. So this is what we figured out. How you do three-factor analysis is that, say this was A, B and C. So in the furthest two genes will have the highest difference in between them, and then you place the third depending what the difference between the other like what is the recombination frequency between each of the pairs are. So that is not really difficult. So we look at it and we know that A and B have a 26% recombination frequency, so they are furthest apart. Then you try to place C somewhere and you know it is 18% away from C, So it could be 18% on the right of right of A, or 18% to the left of A. But then you also know that B and C have a recombination frequency of 10%. So if it was all the way over here, then the recombination frequency between C and D would have been larger than 26%. So it has to be in the middle, and since 10 B and C have a recombination frequency of only 10%, it is closer to B than closer to A. So this is how, if C was your mutant allele, then you have been able to figure out where it lies in the genome between two known markers A and B. And one other point is that this is known as a genetic map. So the blue line is the DNA sequence, and A, B and C are different gene locus. But a genetic map is just imaginary map based on recombination frequency. So you would see that the numbers do not add up because recombination frequency is not an exact thing that separates all the gene loci. So they are not additive. They are estimate of how further the genes are or how tightly they are linked. If you wanted to actually figure out exactly where C is, then you have to make a physical map. A physical map is exact gene sequence or the DNA sequence of this entire stretch. So you know there is a 18 kb difference between A and C and a 10 kb difference between B and C, and then obviously the final distance between A and B will also add up because that is the actual unit that is present on the genome. So genetic map, imaginary map based on linkage analysis on how, like, gives you an estimate of how close the genes are, but the numbers are not additive. Physical map, exact sequence of all the genes and where they are on the genome. Additive, and it is the actual thing that we that is actually present on the genome. Okay, so this is what we got. The allele C is between A and B, is closer to B. Okay, moving forward, what are the advantages of suppressor screens? The options are: it allows for screening of lethal phenotypes of mutant alleles, provides insights on gene-gene interactions, allows heterozygous mutants to be used to determine gene function, allows for screening of gain of function mutants, allows for identification of function of new mutant alleles in other genes. So these are all the options. I will give you another moment to try and figure out what the correct answer is, or in this particular case there are more than one option correct, so you can choose more than one to figure out what the correct options are. Okay, so let us look at it. What exactly are suppressor screens? So say after your mutagenesis screen was over, you have been able to figure out a particular mutant allele and known that loss of function of it creates problems. So say loss of function of the particular allele causes problems in limb development. That's excellent, but how do you then figure out what's the normal function of the gene? So what pathway is it involved in? What other genes does it interact with? What which cells were probably express these genes? So these are the particular questions that you can figure out with a suppressor screen. Maybe not the last one, but the previous two you can figure out, like what pathway it is and what genes it interacts with. So it's a powerful method to isolate additional genes that function in a particular process. So here the example is the multivalva phenotype. So in case of C. elegans, the particular example that we are following is the formation of the vulva. So we had two phenotypes, either multivalva, where there were multiple vulva formations occurring, or vulvaless, which is VUL, in which there was no vulva formation. So say you're looking at a particular mutant allele which causes the multivalva phenotype, and now you want to know which genes does it interact with and which pathway it is involved in. So what you do is that you do a suppressor screen. So you take your multivalva worm. So in the particular mutant, it should have two MUV alleles, but everything else should be wild type. So you put it in EMS all over again and self it, and what you should find is that this one remains the same, because this you imagine is not being affected, but you have a recessive mutation in some other gene, and you then self it again, and again this will follow the Mendelian genetics, and you would end up with two. So this remains the same. So two mutant alleles with multivalva, and you then have another mutation somewhere in the genome which has both the same mutant alleles. So now what you do is that you look for phenotypes in this. So what do you look for? You do not look for like further things that are different from wild type. You try to figure out if this particular new mutation can rescue your previous phenotype. Why is that? This is because then during a suppressor screen, if a further mutation is able to revert back, so which are known as revertant worms. So say you went from multivalva to a normal wild type looking vulva, that means that these two genes interacted somehow, and somehow the mutation in the other locus was able to rescue this. So you are figuring out the genes which interact in the same pathway maybe or somehow to your already known mutant, and this requires, so you can imagine that there is a very low chance of this happening, so you have to screen millions of organisms to actually figure it out. And this is very well done in worms, because like you can actually do it. It is doable in worms. So you do a suppressor screen in worms, and you are able to figure out the genetic interaction. So you look for non-MUV revertants which rescues the multivalva phenotype in F1 or F2 generations. The key is to design the screen such that large number of mutagenized gametes can be observed, because this, as you can imagine, there is a low chance of this mutation actually doing something, and there is an even lower chance that this mutation will revert back or rescue the multivalva, because this has to be that in a specific gene which actually interacts with that, and obviously there are thousands of genes which do not. So very low chance of this happening, and that is why in suppressor screens you have to characterize like millions of gametes. So for an example, suppressor screens were run on the vulva development in C. elegans. So we had two genes that we are interested in, Lin15 and Let60. So after the suppressor screens were done, special alleles of these new genes not recovered in earlier screens, because they have lethal null phenotypes. So for example, Let60, which is the nomenclature for lethal 60, the 60th lethal gene found in that particular mutagenesis screen, the double mutants were lethal, but when a suppressor screen was run, a particular mutation could actually recover the phenotype of Let60, and that let us to know new alleles or new function of these genes, because normal full null knockouts was completely lethal, but partial mutations in the case of suppressor screens, we're able to figure out what exactly it does without actually being lethal so early so that you cannot observe the phenotype. So new alleles of known genes, example let60, several suppressors of lim15 multivulva mutation were new alleles of let60 with vulvaless rather than the original multivulva phenotype. So using this technique, people were able to figure out that lim15 actually interacts with this let60 gene, and since it was a partial mutation between the genes because they were together in the same genome and were able to complement suppress each other, that is when we are able to figure out that these two genes actually function together, and that let60 is just not lethal. Due to lethal phenotype, you are not able to figure out that what exactly its function is, but using the suppressor screen you are able to figure out that it actually plays a role in vulva development as well. So uh, following our example of let60, there are three types of let60 alleles that were figured out using the suppressor screen. A complete loss of function, when you do just the mutagenesis screen and take double mutants, it is lethal. Antimorphic, which is dominant negative, where you have vulvaless, which is uninduced fate of the vulva. And hypermorphic, when it is always active, it is multivulva, which is an induced fate. So you are able to figure out that yes, in the suppressor screen, let60's new alleles come to light, and you are able to figure out its function in vulva development, which is only you are able to do because it is a partial mutation which on top of a different background mutation which helps you to ascertain these particular functions. Normally you would not have been able to. So what are the major advantages of suppressor screens? Allows for screening of lethal phenotypes of mutant alleles, provides insights on gene-gene interactions, and allows for identification of function of new mutant alleles in other genes. Okay, moving on. Yes. Two genes code for enzymes A and B. A loss of function mutation in gene B gives the same phenotype as a loss of function mutations in genes A and B. What are the possible inferences you can make about the pathway? The options are: enzymes A and B are part of a sequential substrate pathway where enzyme A acts before enzyme B. A and B are part of a sequential substrate pathway where enzyme B acts before enzyme A. Enzyme B is epistatic to A. Enzyme A and B are part of a regulatory network where enzyme B activates enzyme A. Enzyme A is epistatic to enzyme B. Enzyme A and B are part of a regulatory network where enzyme A activates enzyme B. So this is more than one option correct kind of a question. So go through it well and figure out what do you think are the correct options. Okay, So let's look at it. So what exactly is epistasis? Epistasis is a technique in which you figure out the sequence in which particular genes act on a particular network. So here we have an example of a sequential substrate product along an enzymatic pathway. So say you are looking at genes that you know act on the arginine synthesis pathway. And so there is a precursor which is then metabolized to ornithine, metabolized to citrulline, and finally metabolized to arginine. You want to figure out what is exactly the order in which these particular enzymes or genes act. So what you do is that you sequentially knock them out. So when you knock enzyme A out, so you have no ornithine forming, only precursor is there. When you knock enzyme B out, you have only ornithine forming but no citrulline. And finally, when you knock enzyme C out, you have only citrulline forming. You have citrulline and ornithine there but no arginine. Now what you do is that you also knock out two at a time. So if I knock out enzyme A also in the second case, what will happen? I will only have the precursor and no ornithine, which is exactly same as the phenotype for only enzyme A knockout. So double knockout shows the exact same phenotype as a single enzyme A knockout. So in this case, I will say that gene A is epistatic to gene B. So whichever single mutant phenotype is equivalent to the double mutant phenotype is known as the epistatic gene. So gene A is epistatic over gene B because double knockout looks like gene A. In sequential substrate pathways, you can see here from the example very clearly is that if the double knockout occurs and it is really similar to the phenotype of only gene A single knockout, then gene A lies upstream of gene B because you only have precursor, but in double knockout again only have precursor, but in gene B knockout you have ornithine, so it has moved forward one step. But without gene A it does not move to ornithine. In double mutant and single A mutation you only find the precursor. So in sequential substrate pathways, whichever gene is epistatic lies upstream. So enzyme A or gene A is epistatic to gene B, so enzyme A is upstream of enzyme B. Same thing you do with these two, because now you know where A and B lie. You now figure it out with B and C. So with B and C, you do the same. You knock out B, you find only ornithine. You knock out C, you only find citrulline. You knock out both B and C, you are left with ornithine again, no citrulline. So with B and the single mutant of B is similar to the double mutant of B and C. So B is epistatic over C, B lies upstream of C, and so you know A lies upstream of B, so the entire pathway is figured out. A, B, like it goes A first, B second, C third. Here there is one point that when people were actually using epistasis to figure out all these pathways, what they did is that what they didn't know is that citrulline comes from ornithine. So you do not know which substrate comes first. So it could have been citrulline here, ornithine here. Then the entire pathway would have been the opposite way around. Or the precursor actually lies is actually one of the middle step and it starts with ornithine. So only with these kind of epistatic analysis can you figure out the particular order of the enzymes as well as what order the metabolites occur in the entire pathway. These were also probed extensively using biochemical methods where they used radio labeled atoms to figure out where what goes, but this is the epistatic method to figure out the order of the genes. So so this was a sequential substrate pathway There is a second kind of pathway known as the regulatory pathway, in which gene A activates gene B activates gene C, and then does some function. In case of regulatory pathways, whichever gene is the epistatic one actually lies downstream. So it is the opposite to sequential substrate pathways. So first thing first, we know that gene B is epistatic to A because single mutation of B is the same as double mutations of A and B. So B is epistatic to A. Now, if it is a sequential substrate pathway, B lies upstream of A, and if it is a regulatory pathway, then then A actually lies upstream of B. The regulatory pathway, it is the opposite way around. So this should also be a correct answer. So figure out which gene is epistatic, the one that has the same phenotype as the double mutant. In case of a substrate pathway, sequential substrate pathway, then that gene is upstream. In case of regulatory pathway, it is downstream. As simple as that. Okay, we move on. Yeah. C. elegans gene X has 3 single nucleotide polymorphisms A, B, and C. The strain is homozygous for SNP-A, is crossed with a strain homozygous for SNP-B. The F1 progeny are crossed with a strain homozygous for SNP-C. If a male is randomly picked from the pool of F2 progeny and crossed with a strain of homozygous for C, what is the chance of progeny having at least one A allele? The options are 100%, 75%, 50%, 25%, and 0%. So I will give you a moment to try and figure this out. Okay, let us look at it. So this exactly is how it is done, how three factor analysis is done nowadays. So previously we saw CN, black, CN, and vestigial, and we figured out how to do 3 factor analysis with all these 3 genes. But nowadays, with the large scale genome wide sequences available, we have figured out a much efficient way of doing it, which is known as like figuring out how to use single nucleotide polymorphisms. The great thing, so what are single nucleotide polymorphisms? So all of us have, like humans have the same uh, genome, but you do know that all, even though we have the same genes, everything is not the same. So there are particular synonymous mutations present in the entire genome, which could either lie in a gene or ORF or lie outside the ORFs, which are just different. There is only a single nucleotide different and does not have any effect on the protein formation most of the time. And these are known as single nucleotide polymorphisms, and our genome is full of it. But in case of lab organisms or lab models such as C. elegans or mice, their genome, they are so purebred that the SNPs are really specific to a particular strain. So the N2 strain for C. elegans will have the same SNPs as any other N2 strain because they essentially have the same genome as compared to us, where every human has different SNPs. So what people figured out is that you can, instead of doing your 3 factor analysis with 3 genes, you can do it with 2 SNPs and a third gene. And since SNPs are much more common and knowing the entire genome sequence gives you the power to actually figure out which SNP is being linked to your particular gene. And so this is the idea, that how the idea is the same, that if two genes are really close to each other during recombination in meiosis, they will not they will be linked together, they will be inherited together. Similarly, in case of SNPs, SNP, even though it is a single nucleotide change, it will be inherited with the same gene if it is tightly linked. And since SNPs are much more abundant in the genome, and you can actually use them to narrow your genetic stretch where your mutation occurs to a really small area. So this is how SNPs are used, and this essentially is a question based on SNP based three-factor analysis. Instead of tracking one gene with two SNPs, we are tracking three SNPs. The first is what we are doing is that we have a strain with SNP A, strain with SNP B, and we cross it. So let us look at how we will do it. So ah, yeah, this should not be written like this. So our uh, first parent would have SNP A on both, so it has both the alleles for SNP A, and it has wild type where SNP B should lie because it doesn't carry the SNP B. Similarly, our second parent will have both alleles for so I will write it this way. So in case of the A SNP, it will have wild type and it carries the B SNP. So it will look like this when we actually figure out the probability of having SNP A in F1 progeny. So we do a cross between these two. So the gametes will be A, wild type, and B. And if you do a Punnett square, you will know that this is a simple Punnett square. A positive, B positive, and you know that the probability of inheriting SNP A is going to be 50% in the F1 progeny, because only half of them will ever have the SNP A. Now this is what we start with, and now what we do, we set up a F1 cross, which will be SNP A and wild type for the second copy, and SNP C and wild type for the allele where SNP A lies. So this will also, yeah, and then should be /C, comma, plus. Yeah, should have drawn the Punnett square here. So this is going to be A positive, B positive, AB, A positive. Yeah, this is fine. So then you have, again when you cross it, the gametes will be A positive, C and positive. So this is only taking the particular, so I don't care if they, so this is either going to be positive or a B SNP type of allele, but I don't care because I only care if it has A or not. So this positive could either so this positive actually corresponds to the SNPC. So in case of SNPC, it has wild type. So from this cross, this is the same as the previous one, only 50% would have the SNP-A in the F2 progeny. So taken together, we have 50% from A and B's cross and 50% from A and C's cross. So taken together, probability will be, since both of these are independent events, just a multiplication of them. So 0.5 into 0.5 equal to 0.25, or 25% probability is there that SNP-A would be SNP-A will be inherited in the F2 progeny. So first cross A and B, you have 50% probability of them having SNP-A. Second cross, again A and C, 50% probability of having allele A, and then you cross with them. cross them to each other, then you have 25% probability of having that the allele A is inherited. Okay, and now you have to make sure that this is going to be heterozygous allele A. Yeah, at least one allele A means that it could also be het for allele A, and that is why you have 25%. If it was like which is homozygous for allele A, then you had to start like that will not be possible. That will be 0% because there will be some mix, not 0%. It will be even less. Okay, go to tab. There is something strange call. I need the call. No, wait, what happened? Yeah. So if you had to have a homozygous for allele A, then you had to take uh, only the progeny which had the SNP for A in the F1 progeny, which will be none because none of them will be homozygous for A. Positive. Now there will be 25% who will be positive for A, and then that will be even diluted here, so it will be 0.25 into 0.25, so 12.5%. But for this question, it is 25%. Okay, you know, draw Punnett squares. Do not be like me. Okay. Next question is, what is the ground state of P and P cells in an uninduced condition? The options are primary, secondary, and tertiary. So, you know, choose whatever you think is the correct option. Okay, so let us look at it. So this is an example of this particular question is from vulval development in C. elegans. So this is everything has now been figured out. So you have various P and P cells which will form the vulva, which is the P3P, P4P, P5P, P6P, P7P, and P8P. People, after looking at it for a long time, figured out that do we have this? Yes, we have this. That these cells are the ones that will form the vulva, but everyone was like, how exactly does it form the vulva? So particular the P6P is known as a primary uh, fate cell, which can form a particular structures just here. Say P5P, P7P are secondary, which will form secondary structures towards the ends, and others will have a tertiary fate, which will form the underlying like everything else around the vulva. So primary, secondary cells, and tertiary cells. Primary forms the major part, secondary forms a smaller part, tertiary forms the part away from the vulva. So this one, so people were like, how exactly does this work? So they did particular laser ablation experiments. So in laser ablation experiments, what they did is that they destroyed particular cells and tried to figure out, yes, this is no, this is not laser ablation, this is like mutation experiments, in which they looked at particular mutants and tried to figure out what exactly happens. Okay. Yeah. So what they did is that they looked at the wild type and they saw that always the P6P cells had the primary fate, P5P or P7P had the secondary, and everything else had the tertiary fate. So they knew that the pattern of cell division was invariant and a particular cell will always know what to do. What they did is that if when they looked at lin26 mutant, which had the vulva-less phenotype, they figured out that no PNP cells were formed. So they figured out that PNP cell production is handled by lin26 and PNP cells are required for forming the vulva. When they looked at lin3 mutation, they again saw vulva-less phenotype, but the reason was different. It seemed that every one of the cells only formed the tertiary field, no primary or secondary, and that is why it didn't have the vulva. So they figured out that it is the lin3 gene which controls the induction of the primary and the secondary fates, and in their absence there is no primary or secondary and thus no vulva formation. So primary and secondary cell types are important for vulva formation. Lin15, which has a multi-vulva phenotype, they figured out that in that case they only have primary or secondary cells, no tertiary cells are formed, which is also important for forming one vulva. And in case of multi-vulva, you have multiple primary secondaries fated cells which form multiple vulvas. So they figured out that lin15 gene is involved in forming the uninduced fate. The tertiary fate is controlled by lin15. It is not there, then everyone is either primary or secondary. So this is what they also figured out, that and then finally they also looked at a let60 dominant mutation, in which in the F1 generation only they figured out that it also found multi-vulva, but with dominant phenotypes you cannot make conclusion, because in case of dominant phenotypes it could be antimorphic, like extra production of a particular gene can have unexpected results. So you cannot draw any conclusion from that. But with this particular example, they figured out what is the ground state of the PNP cells in an uninduced condition. It is the tertiary. So there were also another set of laser ablation experiments in which they were tracing if they deleted the P6P cell. So using laser ablation, they saw that in one of the other cells, P7P or P5P, they will randomly start showing the primary fate. So it just showed that all the cells were pluripotent and could take up any of take up the responsibility of being a primary fate cell if the other was somehow injured or not present. And when they deleted the anchor cell, which actually induces all these primary secondary fates, you saw that all the cells were tertiary fated, which meant that the anchor cell, if it is not there, then it was assumed that without the induction effect or being induced by signals from the anchor cell, all of them go to a ground state or uninduced state, which is the tertiary fate. Okay. Okay, I just heard some sound. Okay, yes. So this is it, and this is the last question for the day. Cosmids, backs, yaks are used to determine the dash map of chromosomes of different organisms. There is only single word answer for it, so I will let you try and answer this. Okay, so we have already gone through this. So back yaks are cloning vectors in which you can clone a large stretch of the large stretch of the genome, and they were used in order to sequence and figure out the physical map of different organisms. So We know what a genetic map is, we just discussed it. It is an imaginary map based on the recombination frequency and linkage analysis. So it's based on the unit centimorgans, and it is a rough estimate of how far genes are. The other one is the physical map, which are made with yaks, cosmids, all these huge, all these vectors which can take huge inserts, and you can then sequence them to figure out the exact DNA sequence of the genome, and you know exactly where each gene is. So genetic map imaginary has imaginary units based on linkage analysis and recombination frequency to give you an estimate where everything is. Physical map gives you the exact DNA sequence via sequencing, so that you know exactly where everything is. So yaks and backs were used to make physical maps of the chromosome. Uh, yes. So this is the last question. Why inferring gene function is difficult from the phenotype of a dominant mutation? The options are: dominance could result in any number of types of changes, dominant mutation always misexpress a gene, dominant mutation cause ectopic expression, dominant mutations are usually toxic. So I will give you a moment to try and figure this out. Okay. So, say for a mutagenesis screen, you did a very nice mutagenesis screen, and you found a mutation which is apparent in the F1 generation. So that is why you had this image here. So it was apparent in the F1 generation. What does that mean? That means the mutation is actually a dominant allele. So even if there is a wild type copy, you see a phenotype. So this is a dominant allele. It dominates over the wild type copy. So but what conclusion can you draw from the phenotype that you observe in the F1 generation? The thing is that you cannot draw much of any conclusions. So this is because inferring functions of dominant mutations are not easy. This is because the dominant mutation can be any of three types. It is not as loss of function when it is just that you just turn off the gene. It can actually have multiple different effects. The dominant mutation phenotype can be due to multiple different effects. First is hypermorphic, which is when the mutation is done, it actually causes hypermorphic mutation, which actually expresses the gene in abnormally large amounts, or causes a constitutively active, always active form of the gene to be expressed. So in that case, well, you can't do much. Like, you do not know the original function of the gene, but you know when a lot of it is present what it does. So that is not the normal function of the gene, obviously, and then you can't figure out what exactly it is. Neomorphic, when there is a lot of that gene, then it might have a new function altogether. So the mutation might infer a new kind of function which is really different from the normal function of the gene. So that's why also you cannot figure out what the normal function of the gene was. Or it could be antimorphic, which is when there is a lot of the gene expression product present, then it does opposite to what it normally did. So again, you cannot figure out what the normal function was. So usually isolation of recessive loss of function alleles is also required, and even though you have isolated that particular gene allele, you have to do a loss of function allele mutation for that particular gene to actually figure out what exactly it does. And this is the example for let60 that we saw, where it had a lethal phenotype, but no, not let60. This is an example for the example for this could be the tau proteins. So in case of multiple mental disorders such as Alzheimer's, the tau protein, what it does normally, it helps in transport of cargo from one end of the cell to the other end. But once there is a dominant mutation of in the tau protein, tau gene, then it has a neomorphic mutation. That is, it has a new function in which it starts to aggregate and form plaques that actually start making a lot of troubles for the neurons and ultimately result in the mental illness. So that is a neomorphic mutation. So with dominant mutations which are evident in the F1 generation, you cannot figure anything out. You need a loss of function recessive mutation to actually figure out what exactly is going on. So dominance could result in any number of types of changes, and you will not be able to figure out what the function of the original gene was. You have to find a loss of function mutation for that gene, allele for that gene, and then you go from there. Okay, so that is all I had for you today. So this is the YouTube link on which all the videos are being uploaded. You can go and check them out whenever you want. Post whatever questions you have in the YouTube channel comments or the forum present on the NPTEL course. And this is the reference for the particular course that is being followed. Okay, then thank you everyone. See you next week. Thank you.